More than 100 years ago, scientists found a mysterious region in embryos called the organizer. Now, a new paper suggests this system may be far older than we thought. To test this, scientists looked at this animal called a comb jelly. Comb jellies are one of the earliest branching animal lineages, so they can give us clues about what early animal embryos may have been like. The researchers did a very delicate experiment. They cut out a tiny piece of tissue from the blastopore lip. This is the tissue around the opening in an early embryo where the body plan is being organized. Then they transplanted that tiny piece into another comb jelly embryo, and something remarkable happened. The embryo started forming an extra body axis. The researchers could see this because the embryo developed extra mouth-like openings. Then they did something even more surprising. They transplanted the blastopore lip into a sea anemone embryo. These animals are evolutionarily very distant. Even there, the sea anemone's own cells started switching on a gene linked to mouth formation in a new place. That means the comb jelly tissue was giving developmental instructions to cells from a completely different animal. The researchers also tested one possible signal behind this effect. They injected mRNA, which is basically an instruction molecule, so the embryo would produce more TGF-beta family signals. When the embryo made more of these signals, some larvae developed extra mouth-like openings. So the bigger idea is this. Before animals existed, single cells could change their behavior over time. But once animals became multicellular, cells also needed to know where they were in the embryo. This paper suggests that organizer-like signaling may be one of the oldest ways cells learn to build organized bodies. Follow for more interesting science content.